everyone. Thank you very much for inviting me to speak. It's lovely to be here today. I've been working in Adrian's bird lab for over 26 years now. So Adrian discovered the MeCP2 protein back in 1990. I arrived in 1997, and I'm going to talk today about a number of key experiments in mice, which have led now to the start of the gene therapy trials for Rett Syndrome. Work in the lab has included much more than mouse work, lots of stuff in cells, in test tubes, but really I'm going to focus on just a few experiments which have progressed the research. And I'm also going to talk about a couple of bits of technology which have been crucial to being able to do this kind of work in mice. So Adrian started off his research on the MeCP2 when he was interested in a particular modification of DNA which happens in all our cells. So here you can see just a little stretch of DNA made up of nucleotides, and these are written down G, A, T, C. And it was found that in the genome, when you get a combination of C followed by G, the C, cytosine, often has a little additional chemical group added to it, methylation. And it seems to be really widespread in the genome, and he was just curious as to why it was there and what it did. So he's, with other people in his lab, set out to investigate this. And this picture, kind of boring picture here, is actually the first sight we had of MeCP2. So this experiment, quite simply, is all the proteins from a rat brain being run out here, sorted by size. And then he took labeled pieces of DNA. One piece was just DNA without the methyl groups, and the other had methyl groups. And he found a particular protein in his jungle of proteins from rat brain bound just to that methylated DNA. Then went on lots of experiments lasting for years and years, cloned MeCP2, cloned the gene, and investigated what was actually in the protein. So MeCP2 is actually quite a simple protein compared to lots of the ones we study. We think it has two functional domains. So one of these is the MBD, methyl binding domain, which is the bit that was binding to the methylated DNA. And then it has a second domain called the NID, NCOR interacting domain, which binds to another protein and brings it close to the DNA so that it can influence expression of our genes. So MeCP2, the name is quite a clunky name to have to say. It's methyl CpG binding protein 2, and there was MeCP2-1, MECP1, but that was kind of not what we worked on that since it was first discovered. Okay, so as I said, MECP2 brings the NCOR complex to the genome, and it's notable that when you look at Rett mutations, most of them actually affect these two important functional domains. So either a mutation which stops them working, or these are nonsense mutations which truncate the protein and you lose the NID. So after we found MeCP2, Brian Hendrick in our lab looked at this methyl binding domain and he then found a whole family of other proteins, known as the MBD proteins, which also have a very similar domain, but they have different additional domains, which mean that they carry out different functions. So when Brian found these proteins, they wanted to investigate what they did, and one of the ways to do this is to actually disrupt the gene and make a mouse with a disrupted gene, and then you look at the mouse and see basically what's wrong with it, and that can help you find out the function of the protein. So I was brought in, I was working in Rotterdam at the time. I moved to Edinburgh in order to help Brian with disrupting these genes or knocking them out. So this is me looking quite enthusiastic. This is actually the room where we actually made the mice. Unfortunately, we're both standing in front of microscopes, so you can't see any of the equipment, but you can see us. And now I'm just going to talk about the technology that we use to make a mouse. So key to doing this are mouse embryonic stem cells. Absolutely crucial technology here. So this is a mouse blastocyst. So it's a mouse embryo 3 days after fertilization. And it's basically a kind of sphere, which is hollow in the middle, but the important part is this kind of clump of cells at one side. You can see them in a diagram here. And these are stem cells, and they can contribute to any tissue in the embryo. At this point, they're all the same, and they will go on to form all the tissues. But we can take the cells from this embryo and we can culture them in a dish. And you can see some just here. And 3 important factors here are that you can grow them in a dish and you can continuously, they will divide and they will retain their stem cell properties. And a feature of mouse ES cells, which isn't found in a lot of ES cells from other species, is that you can manipulate their DNA and introduce complex changes, delete pieces, add pieces, make point mutations, and that's due to an unusually high rate of something called homologous recombination. And we call this process gene targeting. And the last thing is that once you've done gene targeting, you've got your modified ES cells. You can then take the cells, and these little bubbles are ES cells, and then inject them back into another blastocyst, and they will then join the inner cell mass and contribute to a pup, which is eventually born from this embryo. And here you can see a litter of pups. This is the normal black pup you would get if you didn't inject anything into that embryo. And here you can see 2 beautiful pups, we call them chimeras. And the cream colour and the brown fur means that the cells that the hairs are growing from have come from these ES cells and not because they come from a mouse which is cream coloured. And you can then breed these mice when they grow up, and they will transmit your mutation into their offspring. So you then you've made a mouse with your mutation in it. So I'm just going to first talk about our MeCP2 knockout. So this is just where we disrupt the gene. So this is the MeCP2 gene. The important parts are these dark green boxes called exons, and these code for the protein in the genome. So we did gene targeting in ES cells, and what we did was to delete these exons here, and these carry the bulk of the MeCP2 sequence. So then in the genome is and knocked out MeCP2 allele. So ES cells injected into blastocysts, we got nice chimeras, and then we bred them on. And eventually we got male mice, which have, because MeCP2 is on the X chromosome, males just have one, so all of their cells lack MeCP2 protein. We also got female mice, where they have one copy of MeCP2 that works, and this means that the body of that mouse is a mixture of cells, half of which have MeCP2 and half of which don't. Now, at this point, I have to say, during this process, I went off on maternity leave, left the mice to do their breeding, and this paper was published while I was off, which linked MeCP2 to Rett syndrome. So before this, it was just a protein we're interested in, but wasn't linked to any disease. So this really invigorated research, and it became seemingly much more important, not just a kind of academic question of what a protein does. So this heterozygous female mouse is the mouse model for Rett syndrome. But what we want to know is, does knocking out MeCP2 in a mouse do the same kind of thing that mutations in MeCP2 do in people? Because it's not always the case when you make a mouse model. If they did show Rett-like phenotypes, they would then be useful for studying MeCP2 and could be used to test potential therapies. So we looked at our mice. So at first sight, up to about 4 weeks, they looked absolutely normal. They grew, they were weaned from their mother, but from around 4 weeks, in the case of the males, we began to see symptoms appearing. Firstly, just kind of slowing down and walking a bit strange. But the males then progressively got worse, developing a number of phenotypes I'm going to talk about in a moment. And they became sick and died with a sort of median survival of about 9 weeks. And when I say died here, we don't just let the mice die. We use the scoring system I'm going to talk about to judge where they were in the progress of the disease, and they would be euthanized when they got to a point that we thought they might begin to suffer. So female heterozygous mice in the model for Rett, they developed the same kind of neurological phenotypes, but they would progress to a certain level, and then they would stabilize. And these mice appear to have a normal lifespan, and they live with the symptoms they have. So as I said, we developed a scoring system. So firstly, for the welfare of the mice, and secondly, so that we could follow the progress of the disease, and if we wanted to use an intervention, we could see whether it was actually helping. So as I said, the mice slow down, they become inactive, and they will just sit where you put them. The gait is affected, so they have a kind of wide waddling gait, with the feet turned out to the side. They will do this, if you pick them up, they will clasp their hind limbs inwards, whereas a normal mouse would just sort of splay them out to the side. We could also see tremor develop, irregular breathing, and as they got to the end of their progression, you can see just a general loss of condition. The coat becomes dull, eyes kind of dull and partly closed, and just generally don't look happy. And we would score each of these 6 things, from 0 being completely normal, looks like a normal wild type mouse, up to 2, which would be the highest level that they get to. And these we would then add together, and we get this aggregate score, which shows you the progression of the disease. So, in answer to the question on the last slide, both male and female knockout mice do reproduce many aspects of Rett syndrome, including a range of neurological phenotypes, and that's as far as you can model human disease in a mouse. The female heterozygotes, which are actually the true model for Rett, have quite a slow progression and a milder phenotype, and also more variable onset. So most experiments people do with these mice they use the males. And the actual phenotypes we see in the males, we see those things in the females, just they're milder. And we've given these mice out to groups all around the world for their studies on MeCP2, both for basic science research and preclinical studies. So, as John mentioned in his introduction, we wondered whether it was possible to just put back MeCP2 in these mice and see if that would either stabilize their condition or actually reverse it. And this was quite a naive thing to think, really. I think a lot of neuroscientists didn't think this kind of thing would be possible. But we're really not neuroscientists at all, so naive. But we'll just try this and see what happens. So before I tell you about the actual experiment, I want to mention a second piece of technology which is really important for all the following experiments I'm going to talk about. And this is Cre recombinase. So this is a protein from a bacteriophage. It's quite interesting that a lot of the technologies that we use for manipulating DNA in the genome come from microorganisms like bacteriophages, because they're quite good at cutting out bits of DNA and putting it somewhere else, moving it around, and that's part of their life cycle. So Cre recombinase is a single protein, a recombinase acting on LOXP DNA sequences. So what are these sequences? So it's just a little stretch of DNA, it's 34 nucleotides. It has inverted repeats on either side of a central 8 base pair motif. And this motif can go one way or it can go in reverse, and that decides how the LOXP sites actually recombine with each other. And I'm just going to show this sequence as a yellow triangle in the next slides, either one way or the other. And what happens is that you can put these LOXP sites into the genome, into a piece of DNA, using gene targeting. And if you put 2 of them in the same orientation, the Cre will come along, it recognizes these, and it will cut the DNA at both sides. And because of the orientation, the 2 triangles will join together with each other, and you'll end up with one LOXP site left in the genome, and then the bit of DNA in between the 2 is lost. So that's a deletion. If you have them the other way around, it will just flip the DNA into the reverse orientation. But I'm going to be speaking about using them this way in the mouse. So this again is the MeCP2 gene, which I showed for the knockout. And what we did was we took a piece of DNA sequence called stop cassette, which will block, so the gene is transcribed to make RNA and protein in this direction, and putting this piece of DNA to stop production of protein from these exons, which are downstream. So it's essentially the same as the knockout allele, where you don't have the bulk of the protein. So we made this mouse, and the mouse looks like the knockout mouse. But because we've got the LOXP sites, we can then use Cre recombinase to recombine them, and that will remove the stop cassette, and now this has essentially gone back to the gene we had in the first place, the wild type gene. This LOXP site doesn't interfere with production of MeCP2. And the final trick we used was we used Cre recombinase fused to another piece of protein, and this means that the Cre only works if you treat the mice with tamoxifen. So until you treat them with this drug, the gene is off, and then when you treat them with tamoxifen, it's turned back on again. And this means that you can decide exactly when you want to turn the gene back on. So, I've said that. So we can start treatment either before the mice have any symptoms, when they look normal, or we can let symptoms develop and then treat them and turn the gene back on again. So the big question is, can the symptoms be reversed after they've appeared? So the answer is yes. So putting it quite simply, if we treat male mice with stop cassette at 3 to 4 weeks, they never develop the Rett-like symptoms. If we treat them after we've developed the symptoms to quite an advanced stage, the symptoms are reversed and the mice go back to looking more or less like a wild-type mouse. And you can also do this in females. So it was often they were over a year old before they developed their kind of full symptoms, but we can also reverse the symptoms in those mice. So this is put in quite a scientific, dull way, but when we actually did it, it was absolutely amazing to see what happens to the mice. I'm going to show my little videos. This is actually the only video of the experiment I have. So this is a mouse. It's 12 weeks old in this picture here, and he's just sitting in a tray here, and he's really not doing anything. He's not moving. He's not even looking around. You can see, if you look very closely, that his breathing is a bit irregular. And if you pick him up, he does this hind limb clasping, I hope you can see. And we injected this mouse on this day with his first injection of tamoxifen. A few days later, he was already looking a lot better. And this is 4 weeks later. That's the same mouse. He's wandering around. He's quite speedy. I'm not sure if you can see here, but his coat is shinier than it was in the original film. Just generally enjoying life. And so this first mouse, we probably would have decided to euthanize him within the next few days if we hadn't treated him. And then the hind limb clasp is also gone. So there's several kind of significant points from this experiment. But firstly, we now know that MeCP2 is not required for normal brain development. You can tell that from the knockout mice because everything seems to develop normally at first, even without MeCP2. It's just later the brain just doesn't function properly. But once that's happened, if you put back MeCP2, it starts to work again. So all the parts are there. There's no loss of neurons from the brain. They're just not quite working properly. And the biggest thing is that this tells us that there's a potential to cure Rett syndrome if we can put back functional MeCP2, which in itself isn't an easy thing to do, but it means that people are going to and have tried really hard to do that indefinitely. So one final experiment, which is also relevant to therapy, is question, because MeCP2 is in all cells of the body, not just in the nervous system, but is it only really needed in the nervous system, or do you need it in the heart and in the lungs and other tissues which are actually not affected in Rett syndrome? So we did another experiment using exactly the same stop mouse. So here's the gene again. And this time we used a different trick with recombinase in that we expressed it or produced the protein only in the nervous system. So in the nervous system, the stop cassette's taken away and the gene was turned on. But everywhere else, so in muscles and heart and lung and skin, it was still turned off. Oh, you can't see my mouse. It's meant to be grey. So this is a normal mouse that has MeCP2 everywhere in black. This is a knockout which has MeCP2 nowhere, shown in white. And then this is what we call the peripheral knockout, where we have normal amounts of MeCP2 in the nervous system, but not elsewhere. So what we found was that compared to a knockout, which was a stop, this peripheral knockout had absolutely normal survival and very minimal neurological phenotypes. This was done in collaboration with Stuart Cobb's lab and Paul Ross, also looked at heart function, lung function, various things, and they all looked absolutely fine. So, it's barely see any difference if you only have MeCP2 in the nervous system. And this means that if you want to develop a therapy to produce MeCP2 in patients, you only really need to hit the nervous system. So you don't need to worry about treating all the tissues. If you hit the nervous system, that's going to have a major effect. Okay, so in summary, our mouse with mutations in MeCP2, and we've also made patient mutations, so trait mutations. They produce many features of Rett syndrome. So these have been really useful for studying MeCP2 function, both in health and in disease. The reversibility of the Rett-like symptoms shows that cure for Rett is possible. And peripheral knockout shows that we only need to really restore MeCP2 in the nervous system. So what I haven't talked about today is various ways of treating patients. So the current trials are using gene replacement, we put back MeCP2. But there's a number of other ways that are being developed to edit the genome. One of these I'm working on at the moment, DNA base editing, and that's where you go in to the actual gene that has a mutation and then edit mutation back to wild type or back to something functional. So we're at a kind of the clinical trial stage, but things are still happening. There's developments all the time which are going to be useful for treating Rett syndrome. Okay, so acknowledgements. I've got a few people who've been involved in the mouse work, but there's many people who've been through Adrian's lab in the last 30 odd years working on MeCP2. We've also worked with Stuart Cobb's lab quite closely in Edinburgh, and most of our funding comes from Wellcome Trust and the Rett Syndrome research. That's it.